modifications that happen to the RNA and so on and so forth. Here, we will not talk about RNA, we move to the next step, which is protein synthesis. But for protein synthesis, obviously the RNA is read and new amino acids are added. We speak about codons, which is the triplet code, or we also speak about, well, then we're speaking about the genetic code here, okay? The genetic code itself. Guys, is that clear? Are you understanding everything? Class is too silent, I get confused. We all fine, right? Okay. Genetic code. So, when we look into the genetic code, some basic experiments were done by some scientists, and they actually came to some conclusions where they speak about one gene and one enzyme hypothesis. So, if RNA is transferred into protein, they said, scientists earlier said, one gene is involved in synthesis of one protein, or basically one enzyme. Okay? For that, they worked on the Neurospora, which is a fungi. So, Beadle and Totten were the scientists who worked on Neurospora. What they did was, they wanted to see kind of how many genes does the fungi have. How can we identify these? See, basically, guys, I mean, earlier, like 30, 40 years ago, gene sequencing and all this did not exist. So obviously, people did not know how to find genes. The only way they found genes was when something is not working. For example, when some protein is not synthesized, or for example, if something is defective, then they said, if this is defective, then there should be a gene that actually produces some protein which is functional. So only when they found something defective, then they thought that gene actually existed. Until then, they did not know what really existed. Now, one of the ways to find out about the genes, what kind of proteins or enzymes they synthesize, Beadle and Totten worked on Neurospora, which is a fungi. What they did was they took the wild type Neurospora, which is the fungi, and they know that the fungi can easily grow on minimal media. Minimal media is what? The one which does not have a lot of nutrients, basic nutrients. Complete media, we studied earlier, has all the nutrients. Minimal media has the minimum number of nutrients required. So basically, whatever organism that can grow on the minimal media means it can actually synthesize the other compounds using the media. Right? That's a minimal media, minimum requirements needed. What they did was, they know that Neurospora can grow on minimal media. It does not require a lot. Then they took this Neurospora, they did some irradiation. They exposed it to some kind of radiation. They expected some mutations to happen. Sometimes this Neurospora, the fungi, died, but those Neurospora that survived probably had some mutations. How do you know that it has mutations? What they did was, they took this irradiated Neurospora and they started growing it in complete medium. It's growing properly, it's growing kind of fine. Initially, the wild type can grow on minimal, but the irradiated ones, they started growing them on complete media because they did not know what kind of mutation has happened. Once they started growing this irradiated Neurospora on complete media, they took some sample of this irradiated Neurospora and they started plating it on minimal media. Okay? Once you plate it on minimal media, if they don't grow, you now know that some mutation has happened to that Neurospora. Is that clear, guys? Now, see, then they transferred the spores onto minimal media. Fungi with mutations did not grow, obviously. Now they transferred those. So if the fungi is not growing, what they do is they go back to this Neurospora, which is on complete media. So basically, this is irradiated. Now they know it's mutated. They take one more sample from the same plate with the complete media, and they transfer this spores from mutant strains from one complete media to minimal media, and so they took multiple minimum medias and they kind of started adding one nutrient to it. Whichever nutrient the Neurospora responded and started growing, they realized that nutrient was the one which was mutated in the Neurospora. You get that, or is it confusing? Look at this here. They took the Neurospora, right? They irradiated with X-rays. They started growing on complete media. On complete media, it goes very comfortable. It actually can grow on minimal media, but after irradiation, they put on complete media. Now, they took some sample and they put it in minimal media. Now, one of it is not growing. This one, which was transferred, is not growing. But these three transferred on minimal media are growing. So basically, only this neurospora is mutated. It's not growing here. Now they go back here, they take one more sample, and they add it to these cultures. These cultures are also minimal media. These are all minimal media, but the only difference is that they keep adding one nutrient additionally. Different. For example, they have add arginine here. They add lysine amino acid. They add methionine. If it's growing on the methionine, what does it tell you? Which means that irradiation destroyed a gene in the neurospora, which will help the neurospora to synthesize the methionine amino acid. Now they know that, okay, so this sample here, after irradiation has lost the gene which can help in the synthesis of methionine, for example. Is that clear, guys, or is it confusing? Is that clear? All right. All right. So, some other scientists, what they did was they took this even further. Now, because you have irradiated it and because it's not growing on minimal media, but if you add one of the nutrients, it's growing on the minimal media, we know that that gene is modified. Then the question they asked is one of the pathways in this amino acid synthesis destroyed, or are multiple genes in this pathway destroyed? Now, how do you know? So they started adding the precursors. For example, let's say that one of the mutants of the neurospora required arginine, right? Arginine for its growth. So in this irradiated model, that arginine gene is destroyed, so it cannot synthesize the amino acid anymore. So what they did was, they went one step, they actually went 3 steps behind, then they added some precursor. So this precursor is required for ornithine, ornithine is required for citrulline, citrulline is required for arginine. Now we know that the neurospora cannot synthesize arginine. Now the question is the gene for arginine only destroyed, or is the gene for citrulline destroyed, the precursor? Or is the gene for ornithine here destroyed? Are you getting it, guys? So they went back in time to see if this amino acid cannot be synthesized, is the precursor for this amino acid destroyed, or the precursor before that, or the precursor before that. So they added all those precursors in 4 different medias, and whenever they found that, for example, if you add, let's say for example, if you add ornithine here, and the bacteria is growing fine, then you know that the gene for arginine is not destroyed, but the gene for ornithine, which was before, was destroyed. Get that? This way, they try to figure out the pathway as to which of the precursor gene was actually destroyed, not just the direct amino acid itself. Guys, is that clear? Is it confusing or is it clear? So, if this is minimal media, right? Normally they can grow, but after irradiation they are not growing. But if you add arginine to this, now the neurospora is growing very well. So probably you can assume that the gene for arginine is destroyed in this neurospora after irradiation. But to make sure, what they did was, there are few other precursors for example, for arginine. This is one precursor, arginine OCA, let's say POC, and this is arginine eventually. So if they add arginine, it's growing, but then is the gene for arginine destroyed, or is the gene for the precursor for arginine destroyed? So for example, if they added this one, if you add this, which is citrulline, if it's growing, if the neurospora is growing, then it's not about arginine, but this one. Now, they said if they added this ornithine here, if it's not growing, then the mutation actually probably is here or here. Is that clear? So this way they try to find out which the pathway that is affected, not just one of the genes that's affected. So that's how they kind of figured out as what is the biochemical pathway eventually leading to arginine synthesis, and they figured out these steps in Neurospora. Is that clear? That's why they said, but in this case here, step 1, step 2, and step 3 are indications of enzymes. But you have to remember that not all genes are enzymes. Not all proteins are enzymes. Some proteins might not be enzymes, they could be transport proteins. So this kind of research where they try to find out the whole pathway that is destroyed can be applicable for enzymes, because enzymes can actually convert substrates into products. So eventually they said this is one gene, one enzyme hypothesis, but they realized that not all proteins are enzymes, and they also figured out that sometimes some polypeptides can be synthesized by genes. For example, if you take a gene, right? If you take a gene, gene A makes a polypeptide chain that looks like that. Gene B makes another polypeptide that looks like that. And maybe gene C and gene D. So all these together might form one protein, right? So they said one gene probably does not really synthesize an entire protein. It could just be one of the polypeptide chains of a protein. So they changed the concept of one gene, one enzyme to one gene, one polypeptide, indicating that one gene need not really synthesize an entire protein by itself, but could just synthesize one of the polypeptides, and that could be it. You get that, guys? So that's why they changed the hypothesis from one gene, one enzyme to one gene, one polypeptide hypothesis. Is that clear? See, look at this, where I was mentioning. So they know if it's irradiated, look at this data. They said enzyme A, enzyme B, and enzyme C. All these three enzymes are required for eventually for arginine synthesis. Here, if group 1 is blocked at the step, then obviously you need to add this ornithine here for arginine synthesis. If there's a mutation here at group 2, which actually is a mutation for the ornithine, then obviously you need to add citrulline to make arginine. If this is also blocked, then you have to directly add arginine. So this way, when they kept adding one compound, if it's not growing, they said, hey, what is the pathway for the synthesis of this compound? Now let's go back and add those compounds, the precursors, and see if it's actually the mutation within the arginine gene for arginine synthesis, or is it a mutation in a gene that is for the precursors of arginine. So this way they went back in time to figure out, not back in time, back in the cascade or in the pathway to figure out which gene is actually mutated, and they were able to sketch out the whole pathway. And like I said, this technique would work for genes which are enzymes, because enzymes will convert the substrate here into products thereby, but this pathway probably might not work for all the genes, because not all proteins are enzymes, but all enzymes are proteins. I think you know this already. Now, looking at the structure and function of proteins, proteins are made up of amino acids, and we know that amino acids are linked to one another with the help of a polypeptide chain. So if you look at this here, this is one amino acid, and this is the second amino acid. You have the amine group. So amino acids have an amine group and a carboxylic group. That's why they say amino acids can act as acids and bases at the same time. Okay? It has an amine group and a carboxylic group. Here if you see, this is the carboxylic group of an amino acid, this is the amine group of an amino acid. When a peptide bond is formed, usually a water molecule is removed, and then a peptide bond is formed that links two amino acids together. Okay? Peptide bonds link amino acids together. Now the primary structure, looking at the structure of the protein itself, the primary structure of the protein is actually the sequence of its amino acids. The sequence of amino acids in a given protein is referred to as a primary structure. Now this primary structure, so there is some kind of interaction between different amino acids, and they have a pull for one another. So those amino acid chain comes closer, it kind of begins to fold because of the interaction between different groups on a given amino acid that gives rise to the secondary structure. The secondary structure is actually the beta-plated sheets and the alpha helix. The beta-plated and the alpha helix are the secondary structure. Now these beta-plated and alpha helix further fold down to give rise to this tertiary structure. Such tertiary structures combine with one another to give rise to the quaternary structure. So the protein primary structure is amino acid, secondary is beta-plated and alpha helix. Then you have the tertiary structure, which is nothing but the interaction between different beta helix and beta plates and alpha helix. And the quaternary structure is nothing but a further folding and compact structure of protein, referred to as the quaternary structure, shown here in this image. So this is your amino acid sequence, shown here in the image, and this is your secondary structure, the slight folding of the amino acid sequence to give you the alpha helix and beta-plated sheets. This is a tertiary structure. So here they are actually showing the sequence of globin gene, hemoglobin, right? Hemoglobin has 2 alpha and 2 beta globin genes, right? For polypeptides. So this is one polypeptide. When you have 2 alpha and 2 beta, that gives you hemoglobin. So hemoglobin is a very good example of quaternary structure of protein. Primary, secondary, tertiary, and quaternary structures. Is that clear? Proteins have 4 different levels of structure. Now breaking the genetic code. So now, how did they really find out that the nucleotides in an RNA are being translated into proteins? How does this happen? So we know that the set of nucleotides that code for a single amino acid, the basic unit of the genetic code is referred to as a codon. The codon has 3 nucleotides, and that is referred to as a codon. Now, if you imagine that one nucleotide in an RNA is enough to code for an amino acid, then obviously there are only four codons, and you should have only 4 amino acids. But we have 20 different amino acids. Let's assume that the genetic code is a duplet, basically only 2 units, like here, GU, AC, and so on. Let's imagine only 2 nucleotides can give rise to one amino acid. In such a case, you have 4 into 4, 16 possible combinations, but you have a total of 20 amino acids. Then they said probably the genetic code is actually a triplet code. Then you will have the possibility of 64 combinations, but here the amino acids are only 20, and they said probably more than one of the triplet code is actually coding for the same amino acid, which actually happens to be true in the future. So they figured it out. What it means is, if you have 64 different codons, and if you have only 20 amino acids, so more than one set of codon is probably helping, helping the translation of amino acids. So more than one codon is actually carrying the same amino acid. This gives you the concept of something called degeneracy of the code. Understand that? The code is referred, the genetic code is considered degenerate because more than one codon codes for the same amino acid. Now, Marshall Nirenberg and Johann Mithai, actually these were the first scientists who actually worked on something called the polynucleotide. They used the enzyme called polynucleotide phosphorylase. What the enzyme does is, it actually, if you add uracil, if you add adenine, if you add cytosine, whatever you add, it actually links them together. So one of the first experiments that Marshall, Nirenberg, and Mithai did was, they took this enzyme called polynucleotide phosphorylase, they added just uracil to it. Okay? If you add uracil, it's going to add all U's to it. If you put 1,000 U's, it's going to link all the 1,000 U's together, right? Now, what they did was they took this codons, which was generated by this polynucleotide phosphorylase, which is the UUU, and they added to the tube, which had one amino acid in each tube that was radioactively labeled. So let's say there are 20 amino acids. Every amino acid was labeled, but one single amino acid was put in each tube. Now, when they added this triplet codon with all the cell machinery required for RNA or protein synthesis, they found that radioactivity was found in proteins which only had phenylalanine inside it. They were all phenylalanine units only inside it. Now they understood that, all right, if that's the case, UUU codes for phenylalanine. Get that? That's how they, this is the, see, if they had all the 20 amino acids, and then when they added the triplet code, only protein synthesis happened only in the tubes that had phenylalanine, indicating that UUU codes for phenylalanine. You know what's something very funny? This is actually a picture from, this is Mithai, and this is Marshall Nirenberg. Both these guys were German scientists. Very funny, actually. I knew Mithai one-on-one. He didn't complain to me directly that he was not involved. His name was not voted for the Nobel Prize, but he was one of the scientists at the Max Planck Institute where I was working in Germany. I was working in the group of Professor Walter Stummer, and Mithai was actually, he was more or less retired there. There was another department where there was not so much work going there. He had a computer, he simply used to sit there. He was a very tall guy. He was like 70, I think, 70 when I met him, and he was really 6. So probably when he was young, he was 6'4" or 6'5", something like that. This guy here. And this is Nirenberg, and Haragovind Khurana were awarded the Nobel Prize for discovering the genetic code. And Mithai didn't tell me directly, but I heard he used to tell someone that, I was not included for the Nobel Prize. So I talked to him one-on-one. When I talked to him, I didn't know much about him, and then I had some issues with my boss at the Max Planck Institute, the Russian guy. We had a very bad relation anyway. So then he was there, and I went to talk to him for some suggestions. He had one room with just a computer. He was not doing any research. He was already retired by then. Well, of course, he was not involved. His name was not coded in the Nobel Prize for the genetic code. So, well, I think his work was he was so-called forgotten, so to speak. And then he had a computer, and then I talked to him a few times, and then someone said, do you know that, he used to complain to someone that he was working on it. Then I said, what? And I just Googled him up, and then he was actually the one who was the first to use this polynucleotide phosphorylase enzyme to create a UUU and then work and see on the, but later experiments actually kind of decoded the entire genetic code, and he did not do those experiments, I think, and that's why they did not include him. And then he actually, there was another girl from South America, she was from Colombia. I met her, and then she also had problems with her boss, and then she met Mithai to ask for help, and then he said, you know what, let me correct your thesis. He took her thesis, he corrected it, and he was correcting her thesis for months together. She said, these corrections are enough. No, he said, no, more corrections are enough. At a time point, she was fed up. She said, why is he doing this? I'm reaching for help. And then she submitted her thesis and she left. Mithai did not have a lab of his for himself because he was a retired scientist, but I think he loved science so much, he still took the thesis and he kept on correcting it for years together. And she told me once that he created a second thesis out of the first thesis. He made so many corrections. I mean, this is off the subject, but then I knew Mithai one-on-one. He was really tall. I didn't know that he was almost to be awarded the Nobel Prize. And then he didn't have a lab, but then he had a computer, he sat, he did, then he came to Max Planck, he sat, he left, he came, he left, he sat. I knew him. He was 70 years old. This was back in 2008, 2010 years ago. Now this is not, this picture is much older, but I knew him. He was all that hair. But really a tall guy for 70, really a tall guy, wasn't it? But then when I said what my boss was doing with science and how he expects more data, he was like, oh, it's funny, it's fascinating how these scientists these days want to push for more and more scientific data. I understand science is moving too fast and there's so much competition, and he kept speaking on that. I think he worked on that girl's thesis after she left. She was not from his lab. He didn't have a lab. She approached him for some help, and he kept on correcting the manuscript for himself, and he kept himself busy for the next year or two just working by himself on it. it was his own, like a novel, or he just created something out of it. I don't know what he did. Yeah, that's my. At the Max Planck, I did have the luxury of seeing multiple Nobel Prize winners at the same time. I was once drinking coffee, and there was this what's his name? Erwin Neher. He was awarded the Nobel Prize for electrophysiology. I was drinking coffee. He sat just there, like, I was seeing him. I knew him. And then another professor came. They talked some science, and they were like, "Wow, that's a Nobel Prize winner sitting right there in the cafeteria." And he was the only white guy I know. And then every once and every few months, there were Nobel Prize winners invited to Göttingen and to give talks. We used to go and not meet them, but we used to attend the talks. Probably I took life too much for granted, seeing a few Nobel Prize winners. Yeah, Max Planck Institutes are like power play institutes for research, and very powerful people are invited. Anyway, well, okay, so Mikhail and Nuremberg were the ones who actually started using nucleotide phosphorylase to kind of synthesize the triplet codes, and then there are some other steps that they did to go beyond using triplet codes, because for example, if you add uracil, okay, U codes for phenylalanine, probably AAA codes for another amino acid, and CCC codes for another amino acid. But after you have these triplet codes, you also have UAG, UCA. So if you want to have, they further extended the experiments by adding multiple nucleotides and then seeing what amino acids they actually coded for. I'm not going into those talks into that discussion because it's going to get very lengthy here. But so of the 64 possible codons, we know that 3 of them actually are stop codons, right? UAA, UAG, and UGA. These 3 codons do not code for any amino acid, and when the ribosome machinery comes across these triplet codes, either of them, it's actually, since it does not code for any amino acid, termination of protein synthesis happens once these stop codons are encountered. The remaining 61 codons are referred to as sense codons because they code for some amino acid. If they don't code for any amino acid, they are called nonsense codons. If they code for amino acids, they are called sense codons. All right? Sense, nonsense. Since 61 codons code only for 20 different amino acids, which means that more than one codon is coding for the same amino acid, the genetic code is said to be degenerate. Codons that specify the same amino acid are called synonymous codons. So for example, here, if you look at this is the genetic code. If you look at this, CUU, CUC, CUA, CUG, all these actually code for leucine, right? So these 4 codons which code for the same amino acid, which is leucine, are referred to as synonymous codons. Okay, synonymous codons. Now look at this. See, phenylalanine is coded by UUU, UUC, and UUA and UUG that actually codes for leucine. These are the 3 different stop codons, and this is the genetic code. It was eventually kind of postulated by not only Nirenberg but also Hargobind Khorana and who was the other guy? There were 3 people who were awarded the Nobel Prize for cracking the genetic code and saying that these codons code for these amino acids. So this is the tabular column which shows you all the 20 amino acids. This is phenylalanine, leucine, isoleucine, methionine, valine, serine, proline, threonine, alanine. So all these 20 amino acids are mentioned here, and these are the triplet codes that code for the same amino acid. So if you look at it, more than one codon is coding for the same amino acid. That's why the genetic code is said to be degenerate. At the same time, when more than one codon codes for the same amino acid, it's also referred to as, these codons are also referred to as synonymous codons. Okay? Synonymous codons, sense codons, nonsense codons. The genetic code is degenerate. Guys, I need it.